Hi everyone, welcome back for lectures 2 and 3 as part of MLT205 Blood Banking Online. And this unit is going to cover Chapter 3, which is Blood Banking Reagents, Overview and Applications in the Howard textbook. One thing I wanted to reiterate, I guess, before I get into some of the content here today is that the Blood Banking and Transfusion Practices textbook, as you read along with the chapters, you will find at the end of the chapters that they have some multiple choice study questions, and these are really excellent as a means of reviewing the content. So let's begin with our objectives. So firstly, you want to describe the relationship of potency and specificity to blood banking reagents. We will be able to compare polyclonal and monoclonal antibodies. Describe the reagents available for ABO typing. Describe reagents available for D typing. Define the reagent control and describe its purpose. And describe the different types and purposes of reagent red blood cells. Moving on to our second slide for objectives. You will be able to describe the basic principles of antiglobulin testing, distinguish between direct and indirect antiglobulin tests, which we abbreviate as DAT and IAT, and when each would be used. Compare and contrast the composition and appropriate uses of polyspecific and monospecific antiglobulin reagents. Discuss the different sources of errors in the performance of antiglobulin testing, and this refers to false positives and false negatives that we may get in testing. And lastly, our objectives number 3, you will be able to discuss the role of potentiators in immunohematologic testing. Describe the functions of common potentiators in blood bank testing, which include low ionic strength solution, bovine serum albumin, polyethylene glycol, and proteolytic enzymes. Define and identify common lectins used in blood banking, and describe the principles of gel technology, microplate techniques, and solid phase red blood cell adherence techniques. So before we get into different blood banking reagents today, I'd just like to take a few minutes to do a little bit of a review of routine testing that we talked about in our last unit. So we need to remember that with immunohematology reagents, the reagents that we use are used basically to detect antigen and antibody reactions when an antibody finds its target antigen. We are able to tell when this happens in vitro by observing either agglutination or hemolysis. The methods that can be used for blood bank testing, traditional method is with test tubes. There's a gel technology, and then a later technology is called solid phase adherence. So just a little bit more about our testing review. It's really important that everyone understands the relationships between antibodies and their target antigens. So all routine methods in blood banking use a source of antigen, and remember this is going to be a red blood cell, okay? And so remember, antigens are markers simply that are embedded in the surface or the phospholipid membrane. They're phospholipids in the membrane of the red cell. And a source of antibody, which is going to be an antiserum or a human serum or plasma to detect agglutination or hemolysis. So this picture may look familiar from our last unit. Basically here we have a red cell, and it has some antigens that sort of look like little knobs around its surface. And when we combine these red cells with an antibody that is directed against them, what happens, remember, are 2 stages of agglutination reactions, right? There's sensitization, where the FAB binding sites of the antibody bind the target antigen and latch on, okay? And then the second stage is lattice formation. And so we see how, nicely, I would ask you guys, if we were meeting face to face, what antibody is this big one? And hopefully you would tell me IgM. So we could see how some of the binding sites of this IgM are attracted to an antigen on one red cell, and then other binding sites of the same IgM molecule are crossing over to an antigen on a different red blood cell, and this is how we are able to see agglutination through that cross-linking and lattice formation. So let's talk a little bit about our blood banking reagents. So there are 4 basic categories of reagents. Firstly, we have red blood cells that come from a manufacturer with known antigens. So the company basically tests these red cells and sells them to us, and we know which antigens are present on the red cells. So we call them reagent red cells. There are also antisera with known antibodies. And so here we see a picture from our textbook. This is antibody against A antigen, antibody against B antigen, and antibody against the big D antigen. Okay, these are known antisera. There are also antiglobulin reagents. These are antibodies that are actually directed against human IgG and or human complement. And then lastly, potentiators are reagents that we use in our test system to enhance antibody and antigen interactions in some way. Okay, so we'll be overviewing all of these today. So as far as reagent regulation, commercial blood banking reagents are licensed by the Center for Biologics Evaluation and Research, which is also known as the Food and Drug Administration, the FDA. The FDA's criteria for how they regulate reagents can be found in something called the Code of Federal Regulations. All reagents must meet minimum standards before being licensed. And 2 terms you see listed here are very important with regard to our reagents. So firstly, specificity describes the ability of an antigenic determinant to be found or recognized by its corresponding antibody. Okay, so that's the specificity. The potency relates to the strength of the reaction when that antibody finds its target antigen. Okay, and so in the last unit, we talked about how in blood banking, when we have positive reactions, these reactions must be graded 1 + 2 + 3 + 4 + 4 + being the strongest. So potency describes strength of the reactions that we get with our reagents in testing. Just like other areas of the lab, quality control is essential to obtaining accurate results when we're doing our testing in blood bank. And so each lab has a quality control program where all reagents, equipment, and procedures that are going to be used for patient results in any given day must be tested to ensure that we are getting accurate results. Components of a QC program include a statement of the criteria for acceptable reagent performance, documentation of reagent use, writing down lot numbers, expiration dates, and the reactions obtained when QC is tested. And as you all have learned in other classes, specifically hematology last semester, when QC is out of range, corrective actions must be taken, okay, to determine why that QC isn't working properly. And we cannot report out any patient results until we achieve optimal reactivity with our quality control. Commercial antibody reagents that are used in blood banking can be monoclonal or polyclonal. And so on this slide, we're going to talk a little bit about polyclonal. So polyclonal antibodies, you guys recognize that prefix poly refers to many. And so this reagent is made from several different clones of B cells. And maybe you recall, B cells are the ones that transform into plasma cells that make the antibodies. Well, here we have clones of B cells that secrete antibodies of different specificities. Okay, and so polyclonal, we have our different B cells here, and they're making different antibodies. Okay, so they could recognize multiple antigenic epitopes. Okay, remember, an antigen can have more than one piece to it that an antibody is directed against. And an example of a polyclonal antibody that we'll be talking in more detail about later in this unit is anti-human globulin, which you will see abbreviated as AHG, anti-human globulin. And so anti-human globulin that is polyclonal contains an antibody against human IgG and also an antibody against human complement C3. Okay, so hence it's polyclonal. Monoclonal antibodies, on the other hand, are made from a single clone of B cells that secrete antibodies of the same specificity. And this is a picture from our book where they basically show how they get these hybridomas that are producing the antibodies, and they're monoclonal antibodies. So monoclonal antibodies are going to recognize one single epitope. Okay, and an example would be anti-A or anti-little c or anti-IgG antibodies. So they're just recognizing that one thing. And so whenever you see abbreviated anti with a dash, this means antibody. So this is table 3.1 from our textbook that basically gives a summary and compares monoclonal and polyclonal antibodies. So, not too difficult. Monoclonal antibodies, remember, are from a single clone of B cells, whereas polyclonal are from several different clones of B cells. Monoclonal antibody is going to be directed against one specific epitope, and it's only going to be one immunoglobulin class. It will either be IgG or IgM. Polyclonal could be a mixture of both IgM and IgG antibodies and contain multiple antibodies recognizing different epitopes of the same antigen. And so if you don't remember from our last unit what epitope means, this is a distinct portion of an antigen, which is the target of the antibody that will combine with the antibody. So a specific piece that the antibody is directed against. So we're going to move on next and talk about ABO typing. And so anti-A and anti-B reagents, and remember, these are antibodies against A antigen, antibodies against B. They're reagents which sometimes in the lab people will refer to as anti-sera. They are used to determine the ABO blood type of a patient. And so basically, we make a dilution of our patient's red blood cells, and in one tube we add a drop of anti-A, and in the other tube we add a drop of anti-B, and we want to see if agglutination occurs. Okay, so if we get agglutination with anti-A, it means the patient has A antigen because that antibody against A found its target, and we're seeing the red blood cells agglutinate. Likewise, if we see agglutination with anti-B, that would mean that the patient has the B antigen on their red cells. And so we use these known anti-sera to find out the unknown, which antigens are on the patient's red cells. So this is table 3.2 from our textbook, which talks about red cell testing, and this is called ABO forward testing. And this is a nice summary, and we'll be going over this in our labs as well. So anyway, getting back to this table 3.2, what we see here in our first column are different blood group antigens of the ABO system. And we see A, B, AB, and O. And then what we see are anticipated reactions for each one of these blood types with different anti-sera. Okay, so when we have an individual that has the A antigen, we should see agglutination reaction with the antibody against A. Because remember, this anti-A is going to find its target, A antigen, and we're going to see the agglutination in the tube. If the person does not have B antigen, okay, and only A, we wouldn't see any reaction with antibody against B. And so the phenotype would be type A. Okay. When we have an individual who has the phenotype of B, blood group B, they have the B antigen on their red cells, and that's the only antigen they have. So do we expect their red cells to agglutinate with antibody against A? No, because they lack the A antigen. But we would expect to see agglutination with the antibody directed against B. When we move on to our next one, phenotype AB is the rarest phenotype of all. And these individuals have both the A antigen and the B antigen on their red cells. And very, very small percentage of the population is type AB. So since they have both A and B antigens, we would expect to get a reaction with anti-A, antibodies finding the A antigen, and then also a reaction with anti-B. Okay, it's finding the B antigen. And then the last type, O, basically think of it as 0. That this is the blood group that lacks both the A and the B antigen. And so basically we are not going to see reactions with anti-A or anti-B because those blood group antigens are absent. And so you may know that among all races and ethnic backgrounds, group O is the most common blood group. And because type O lacks both A and B antigens, it's called the universal red cell donor. Okay, so what this means is if someone was in need of an emergency transfusion and we didn't know their blood type, we could always give them O red cells that wouldn't cause any harm to anyone. So looking more closely at ABO reagents, these reagents are designed by the manufacturer to give us strong reactions, 3+ to 4+ agglutination reactions. And if you recall from our last unit, we talked about how 3+ and 4+ would look in a test tube. 4+ would be one solid mass of red cells that comes off of the bottom of the tube. 3+ would be maybe 1 or 2 big pieces of red cells that come off of the tube. And they are designed this way to reduce errors in interpretation. Okay, so blood typing is critical. Okay, it's critical that we don't make mistakes. And there are a couple of other things that are built into the system with these reagents. So when we're looking at our antisera that we use to determine the antigens that our patient may have, universally throughout the world, anti-A, antibody against A, has a blue dye in it, and this is to reduce mistakes in interpretation. Anti-B is always yellow, okay? And that's intentional, all right? So basically, you know, when we do this testing, we have a couple of test tubes, and so we would need a minimum of 2. One for our patient labeled anti-A, the second one labeled anti-B. 1 drop of the anti-A goes into the A tube, 1 drop of anti-B antisera would go into the B tube, and then we add a drop of our patient's diluted red cells, 1 drop into the A tube and 1 tube into the B tube, and then we centrifuge them immediately for 15 seconds. And this is called immediate spin testing. So immediate spin means as soon as I add everything in the tubes, I give them a little shake, I put them in the centrifuge, I centrifuge them for 15 seconds at 3,500 RPMs, and then I read them with my viewing lamp and mirror to determine if I have agglutination, and if I do, I'm going to grade those reactions, and we expect them to be pretty strong. And so that's what we mean by immediate spin testing. It takes place at room temperature. In blood bank, we also do confirmatory testing called reverse typing, where we look for expected ABO antibodies in our patient. And I'll be talking more about this in our labs for this unit. But basically, what this means is, and you'll be hearing this again from me, and I guess you can't really hear it too many times, but around the turn of the century, maybe the 1900s, Carl Landsteiner basically discovered that we human beings have ABO antibodies to the antigens that are not present on our red blood cells, okay? So what does this mean? I will tell you, I am type A, and so you may know by now, that means I have the A antigen. But due to Landsteiner's rule, I also have an antibody against the B antigen because I don't have the B antigen, okay? And so we'll be talking in more detail about this, but those are called ABO antibodies and they're naturally occurring. So other blood bank reagents that we use in our initial ABO and RH typing include antisera to detect the big D antigen. And the big D antigen is part of the RH blood group system, which we will study more closely in Chapter 6. There are other antigens that are part of the RH system besides the big D, but the D antigen is really the most important for blood bank and transfusion medicine because it is extremely immunogenic. What that means is the big D antigen, when introduced into someone who lacks that antigen, is very capable of stimulating that person to make an antibody against it, okay? And so we'll be getting into details about you know, what we do in cases of transfusion with regard to this big D antigen later, okay? But just know for now that when an individual has big D antigen, we call them RH positive. When an individual lacks the big D antigen, we call them RH negative. Okay. And then AABB is the American Association for Blood Bank. They put forth standards that are really followed throughout the United States. And AABB requires that all blood samples be typed for the big D antigen. So what that means is it's not good enough just to know somebody's ABO type. Oh, I'm type A, B, or what have you. We also need to say, oh, I'm A positive or I'm B negative or what have you. So for our D antigen typing procedure, similar to what we talked about with anti-A and anti-B, there is a commercial antiserum directed against D antigen. And in the same way that we do our forward typing, we would have an additional tube. I would label it D, and I would add a drop of anti-D with a drop of my patient's diluted red blood cells. And centrifuge at immediate spin, just like we previously discussed. And if I see agglutination, I'm going to grade it 1+, 2+, 3+. However, if there is agglutination, as I said to you guys on the last slide, it means that individual is RH positive. They have the big D antigen on their red cells. If there is no agglutination, we suspect they do not have the D antigen and they are most likely RH negative. When we're doing this D antigen testing, some labs will also incorporate something called a reagent control when they are doing the D testing. And the reagent control is really done to make sure that reactions with this anti-D are not due to a false positive reaction. Okay? So let's look at table 3.3 briefly to try to figure out what this means, okay? So here's the thing. Whenever we have an antiserum, like our anti-D, that we get from a manufacturer, this is not 100% of antibody against D. It's diluted with a certain type of diluent, such as albumin, okay, or something of that nature. And so we want to make sure, specifically with anti-D, that an individual is not reacting to the diluent that goes into making the antibody, okay? And so the reagent control is basically the diluent only that would be used in diluting the antibody against D in the reagent. Okay, so just to reiterate, let's say our anti-D is diluted to some point with albumin. This would just be albumin, the reagent control. Okay, so when we test the anti-D tube and the reagent control, when we see agglutination in the D tube and no agglutination with the albumin alone, that's good. Okay, so what that means is that the agglutination that occurred in this tube is truly due to the patient reacting with the antibody against D. They're not reacting with albumin diluent. Okay, when someone is truly negative, we would expect to see a negative with the anti-D and a negative with the reagent control. Okay, no problems there. Now, when we run into trouble, and the whole purpose for doing this testing really is when we see a person react with the anti-D, but also with the reagent control, which is basically just the albumin diluent by itself. We cannot interpret this. We don't know if this patient is Rh positive or not because they're reacting with the diluent, and that could be the cross-reactivity or the carryover that we're seeing here in the D tube. Okay, so what do we do in a case like this? Most often, a lab will then go to a different anti-D formulation that maybe uses a lower percentage of albumin as the diluent, and maybe that cross-reactivity disappears. Okay, so with the anti-D reagents, some are prepared as a high-protein reagent. And so they contain polyclonal antibodies with about 20% bovine albumin. And these high-protein reagents are the one that we can sometimes see false positive agglutination with, where that reagent control would be very important to run alongside. Okay. Newer formulated low-protein reagents have monoclonal IgM or monoclonal and polyclonal blends. But what's really important here is they use a much lower percentage, only 6% of albumin, and so they're less likely to show cross-reactivity. Okay, so when we're using a low-protein reagent, there's no need to run that reagent control. So let's just go back to that slide. So if we were using the high-protein reagent, 20% albumin reagent, we would need to run the reagent control. Okay, and if we saw a situation like this, where the patient is reacting with both, and we're not sure then, are they really D positive or not, we're going to retest them with a low protein reagent. And it should minimize those false reactions that we see with the high protein reagents. So this is our synopsis of the low protein reagent controls. So our reagent controls basically ensure that our typing results are correct and should not agglutinate. When do we get false positives? When there are protein abnormalities, but also sometimes patients have autoantibodies, and these are antibodies directed against self. That's what that prefix auto means. And they're called cold autoantibodies because they react at room temperature. And so they could be cross-reacting with our tests that we're doing at room temperature. So when do we use a separate reagent control? If red blood cells agglutinate with all of our ABO antisera, we're going to suspect false agglutination reactions. And if there is this polyagglutination going on, the reagent control is probably going to agglutinate too, and that's going to tell us that something's going on. They're not needed okay, if we have a negative result using any of the low protein reagents. Okay, so we're going to move away from anti-sera for now, and we're going to get into a little bit of detail about our reagent red blood cells. So just like we have known antibodies that come from a manufacturer, manufacturers can also have reagent red blood cells for us. And they're going to be used in the opposite way. So our reagent red blood cells, remember, they have antigens on them that are known, okay? We're going to use these to determine which antibodies the patient may have in their serum or plasma. Okay, so the known here are the antigens, and the unknown is, which antibodies does our patient have? So uses for our commercial red blood cells, ABO reverse typing. Okay, so a few minutes ago when I was discussing with you guys about LynnC's rule, how reverse typing tells us which naturally occurring antibodies a patient has of ABO variety, we would use them for that. We can also use separate reagent red blood cells called screening cells to test for unexpected antibodies. So maybe when you were doing your phlebotomy rotation, you saw some types and screens drawn. So the type is the ABO and the RH type. The screen is the antibody screen. And so these are reagent cells that are going to tell us if a person has formed an antibody from a prior transfusion or in a woman who's had a pregnancy, okay? And so basically what this means is a person is stimulated by red cells from another person, whether it's donor red cells here or fetal red cells here. They get into the circulation and the individual is stimulated to make these unexpected antibodies. Okay, we use screening cells to find out if they have any of them, okay? Now, if we find out, the screening cells are reacting, our patient has an unexpected antibody, our work isn't done, we then need to go on and determine the identity of the antibody, and we use other reagent red cells, in this case called panel cells. And this testing is really kind of fun. If you enjoy solving puzzles, you will like this. For now, we're just really doing an overview, okay, just so you have an idea of all of the reagents we use. So let's talk first about our A1 and B cells. Okay, and here we have table 3.5, which is showing reverse typing results based on our person's phenotype or their antigens that they have. Okay, but before we do that, let's talk about how this testing is done. Okay, so we would have a separate tube for A1 cells and B cells. And first, we're going to put 2 drops of our patient's serum or plasma into each one of these tubes. And so when we are looking for antibodies in our patient's serum or plasma, we always double up the volume to 2 drops. So you may recall when we were looking at the anti-serum a couple minutes ago, when I said it's 1 drop of anti-serum and 1 drop of cells. Well, anti-serum, remember, are designed to react strongly, so they're more potent than perhaps the level of antibodies that a patient would have in their serum or plasma. So once we have our 2 drops of our patient's serum or plasma in each of the tubes, we're going to add 1 drop of A1 cells to that tube and 1 drop of B cells to that tube. And we're going to do our immediate spin testing in the same way. We're going to mix those tubes up a little bit, put them in our centrifuge, and centrifuge for 15 seconds at 3,500 RPMs. And the same rules apply. We're going to look for agglutination. We're going to grade the strength of the reactions. And LynnC's rule is going to help us figure out if it makes sense. Okay, so remember, here's LynnC's rule again. All of us have naturally occurring antibodies to antigens that are absent from our red cells. Okay, so Think about it. Would it make sense for me to have an antibody against an antigen that is on my red cells? I would be agglutinating my own red cells, correct? So with ABO, these are really one of the only naturally occurring antibodies. We have antibodies to the antigens lacking from our own red cells. Okay, so let's look more carefully at that. So here we have type A. This person has A antigen. So we're going to reverse our thinking. Okay, we want to say, okay, well, what antibodies would a person who is type A have? They would not have an anti-A. Okay, an anti-A would be the one that would react with A1 cells. Okay, they'd be reacting with themselves in that case. We expect them to have an antibody against B antigen because they lack the B antigen. And so remember, these reagent B cells have B antigen on them. So these are the cells we think they are going to react because they have naturally occurring anti-B. Okay? So person who is type B has the B antigen. We expect them, remember, to have an antibody to the antigen they don't have. We expect them to have anti-A. Okay, so their anti-A is going to react when it comes into contact with our reagent A1 cells that have the A antigen. We do not expect them to have anti-B to react with B cells because they are type B. Okay, so they're going to have anti-A and react with A1 cells. The rarest blood group, type AB, these individuals have both A and B antigens. So guess what? Since they have both of these, they have no antibodies. We would get negative results with reagent A1 cells and negative result with reagent B cells because they lack both anti-A and anti-B. Okay? And then lastly, type O has no A or B antigens, so we expect them to have both anti-A and anti-B. We expect them to react with both the A1 cells and the B cells. Okay? So that is the basis of reverse typing. So just remember, forward typing, we use our known antisera to determine the antigens the person has. Reverse typing, we use our known reagent red cells with known antigens to determine which naturally occurring ABO antibodies the person has. And the two should agree. It's a double check, which is why we do both. You don't want to make a mistake in somebody's blood type. Someone getting an incompatible blood type, transfusion could be dead within a few minutes of that transfusion. It's very, very serious. So doing both forward and reverse typing reduces the chance or the likelihood of mistakes. Okay, so our reagent A1 and B cells can be from a single donor or a pool of donors when the manufacturer makes them. They are prepared and come to us at a 2 to 5% concentration. This is not straight whole red blood cells packed together. Okay, think about in our last lecture how we talked about the zone of equivalence. Remember that one? So we're going to have optimal reactivity for visualizing immune complexes and agglutination when our antigen is in about the same concentration as our antibody. So with blood bank testing in the tube, 2 to 5% red cell concentration is what we want to have for our testing. These reagent red cells must lack Rh antigens. We don't want the big D or other Rh antigens, C and E, that we'll talk about later, interfering with us getting the right ABO type, okay? Because when they're present, they could be reacting, okay? And our reagent red cells should always be healthy red. They should not be dark or clumped or be hemolyzed. Otherwise, we're not going to get optimal results. Oh yes, the good old days. So this is a picture I took in student lab I miss student lab. Oh, I wish we could be together to do student lab. Anyway, this is a picture I took of our reagents, and so, you know, as we talked a few minutes ago, anti-A, you note the color, commit that to memory. So A is always blue, B is always yellow, and here we see our A1 cells and our B cells. So it's very important to read your labels, okay, because all of these cells can look very similar. What you can't see on the label is it says 2 to 5% suspension, probably where it's curved around. But, you know, before we add any drops of these cells to our tubes, we absolutely have to make sure that they're well mixed, and that the dropper, we have to squeeze it up and down and mix what's in the dropper as well, okay, because we want to make sure that all of those antigens, we're getting the proper antigen concentration when we're doing our test. So let's move on and talk about some other types of red cell reagents. And these are the screening cells. So as I mentioned to you all a few minutes ago, type and screen is the most common test done in blood banking. And we talked about ABO and RH type, and this is the second part, the antibody screen. So these are cells we use to detect unexpected antibodies in a test called the antibody screen. We are not trying to detect ABO antibodies here. Remember, those are expected, and we know what to expect based on somebody's type, right? So these are the antibodies that a person may have formed from prior transfusion or pregnancy when they were exposed to the red cells of another, whether it's a donor red cell or a fetal red cell. So screening cells come to us in sets of 2 or 3 vials. Each vial is from a type O donor. Think about why that's important. It must be type O, because O lacks all ABO antigens, right? We don't want any other type, because then technically if an individual has anti-A or anti-B antibodies in their blood when we do the testing, could be reacting. Okay, so must be type O, could be from a single donor or 2 donors pulled together. So pooled cells are only acceptable, they can be used for donor testing. So if you were working at the Red Cross or a blood donor center, they could be used there. But when you're working in a hospital clinical lab, we can only use single donor vials, okay, to test a person who's about to receive a transfusion, and that person is known as the recipient. So the single donor cells tend to give stronger reactions, and you know, we don't want to make any mistakes here, so very important. So moving on, some other information about our screening cells. Each lot of screening cells comes from the manufacturer with something called an antigram. And the antigram shows us which antigens are present or not present on that vial of cells. Okay, and so this is a picture from our textbook of an antigram. And when you look across here, these are other blood group systems that we will be talking about a little bit later in module 2, I believe. So there's the RH system, there's MNSS, P1, Lewis, Lutheran, Cal, Duffy, Kid. Remember, we're not using screening cells for ABO. Okay, we're using them to find antibodies to antigens of these other systems. Okay, and so here we see that these screening cells, there's a cell 1 and a cell 2. And in module 2, we'll talk about what these R's mean. Okay, don't worry about that for now. But when we look at screening cell 1 and we go across, we could see whenever there is a plus sign, it indicates that this cell has that antigen on it. So we could see that screening cell 1 has the big D, it has the big C. Okay, when we move on to E, it's a 0, it means it lacks big E. It lacks little c. It has little e. Doesn't have F, doesn't have CW, has both M and N. Okay, so you get the idea, so on and so forth. These screening cells are put together in such a way to try to have different representation between the cells. So what does this mean? If we look at screening cell 2, for instance, we see, okay, well, it also has big D, aha, but it lacks big C. Okay, whereas screening cell 1 had it. It has big E, whereas screening cell 1 did not. Okay, so we want to try to have, this different varying concentration of antigen representation so that when our patient reacts with one of these cells, we can figure out, okay, basically, what their antibody might be directed against. Okay? So these cells must express antigens associated with the most clinically significant antibodies. And so these systems here are the ones that are considered, clinically significant. They're also regulated by the FDA, and so these are the antigens that must be represented on at least one of the screening cells. And so if you look through, you're going to see, okay, that they are there. And we'll be talking more about these systems, as I said, in Module 2. Haha, so here's a picture of our screening cells from last summer. They use Roman numerals. So you see Roman numeral 1 and Roman numeral 2. And this is the antigram that came with them. Okay, maybe it was from the year before, 2018, 2 years ago. Okay, but we could see screening cell 1. This one has these antigens. That's its antigram. Screening cell 2 has these. Okay, and we're basically testing our patient's serum or plasma, 2 drops in a tube with screening cell 1, 2 drops in a separate tube with screening cell 2. And, we want to see if there's reactivity. And if there is, then we need to find out exactly, what the patient is reacting to, which antigens. Okay, so screening cell is just going to tell us yes or no. Does our patient have an unexpected antibody? If we get negative reactions with each screening cell, then no, they don't. If they react with even one of them, then yes, they do, and we need to do more work to find out what antibody or antibodies they may have. So when we have a person with a positive antibody screen, we move on next to the panel. And we use things called panel cells to help figure out the identity of the antibody. Okay? So these are reagent red cells. They're very similar, to screening cells, except a panel has 10 or more different cell vials. Once again, it's critical. They must be type O because we don't want any cross-reactivity with ABO antibodies. So they would be used to help identify the antibodies, unexpected. Excuse me, got to take a drink of water for 1 minute. My allergies are bad today. To help identify antibodies that a person has, and these are unexpected, and it's called a panel test. So just like each lot number of screening cells has an antigram, each lot number of panel cells also has an antigram that shows us what antigens are represented on each vial of cells in the panel. Here we go. Might look a little crazy. So this is, I may have taken this picture. Maybe it's from our book. I'm not sure. But anyway, this is the antigram for a panel. And you might say, oh my gosh, that looks crazy, right? So here, these are the number of vials of cells. There are 11 in this one, okay? And when you go across this way, you could see which antigens are there or not there for each specific panel cell. So let's look at panel cell 1. No big D has big C, no big E, so on and so forth. And you can see how they have different representation throughout, okay? When we have slashes here, we don't know, okay? But when we get into panel testing later, just remember, we're going to test 11 tubes, 2 drops of our patient's serum or plasma into each tube, and then 1 drop of the corresponding panel cell into the tubes. And we're going to use the pattern of reactivity to determine the identity of the antibody or antibodies that that patient has. And yes, sometimes patients can have more than one. They've had multiple transfusions in the past. It's fun. It's kind of like doing a puzzle, but we'll talk more about that later. So here we have a picture of one that we had from 2 years ago. And so you could see how each panel cell is numbered 1 through 10. I think this is called TC. Yeah. So basically 11, okay, 10 plus the TC. And, you know, each antigram is going to be different coming from the manufacturer, okay? And we're going to test our patient with each one of these cells, and it's going to help us determine the identity of their antibody or antibodies. So the next concept we're going to talk about is the antiglobulin test. And this test is kind of one of the more confusing topics of blood banks, so bear with it. I'll do my best, and you may have to listen to it more than one time or start discussion on our discussion board in Canvas about it, because sometimes it can seem a little confusing until you get it. So the principle of the test, it's a commercial antibody that has a specificity towards human globulins, okay? And it's used to agglutinate antibody-coated red cells, okay? So we're going to be talking about other types of tests down the road where a red cell has an antibody attached to it. And so here's a picture from our book where we could see, you know, that these green antibody molecules are attached to these red cells. And so sensitization has occurred. However, the antiglobulin test comes into play because we may not be able to see that sensitization occurred, okay? So imagine for a moment that you're just picturing this red cell here without the white immunoglobulins here, okay? Just the green ones attached. And we see that, you know, the binding sites are attached here to only a single red cell. There's really no cross-linking or lattice formation. And so although sensitization has occurred here, you know, if we were just going to stop there, we wouldn't be able to visualize agglutination. So antiglobulin reagents basically help us to determine if this sensitization has occurred, meaning antibody has bound to its target antigen. And that's their job, okay? So antiglobulin reagents contain antibodies that are directed towards human IgG. Okay, here's where you have to wrap your head around it. It's an antibody against human IgG, okay? And or an antibody directed against human complement, which is usually C3, so either anti-C3d or C3b. So how do we get these reagents? Let's think about it for a minute. And let's see if I can get my pen going here. I might have to draw something if I can. Where's my pen? I'm not finding it. Draw. There's my pen. Okay, so let's say we have oh, that's not going to work. I wish I was meeting you guys in class. It would be much easier. Let me just draw a line. Let's say no, it's not going to work either. Sorry, guys. You're just going to have to visualize it, and I'm going to post some other. There are some other handouts that will help with this. Okay, so they basically inject. Let's say we have a rabbit or a mouse, and we inject that rabbit or mouse with human IgG. So think about it. In the rabbit or the mouse, when it's injected, with human IgG, the rabbit or the mouse says, hey, that's not mine. That's an antigen to me, right? That's foreign. And that rabbit or mouse is going to make an antibody against what's been injected into it. In this case, that happens to be human IgG. So hence, that's how we get antibody directed against human IgG. Okay, we inject human IgG into another animal. It's recognized as something foreign, and the animal makes an antibody against it. Okay, and we call it anti-IgG. So let's look at our picture here for a moment, and let's see what happens here. Okay, so when we have, our original sensitization with the green antibodies attached here, we're not able to visualize it. When we add our antibody against IgG into our test system, this anti-IgG says, remember, it's an antibody directed against IgG, so its target, its antigen in this case, is the IgG. It says, hey, there's my target, and it links on to these human IgG molecules. And then look and see what happens. This is a great picture down here. Now we're getting cross-linking, right? So the anti-IgG links onto it, a human IgG here, one target, one of its binding sites here, and now we're getting cross-linking. So now we're able to visualize through agglutination, we're going to get this lattice network. We're able to see that this sensitization occurred. Okay? So these antiglobulin reagents are tools that we use in our toolbox to help detect sensitization that we may not be able to visualize. Okay? All right, so similarly, antibody against complement, human complement would be obtained by injecting an animal with human complement. And the animal's going to say, hey, that's not my complement, that's something foreign. I need to make an antibody against it. And so hence anti-complement. I hope that makes sense to you guys. Okay, so let's talk about the reagents. Some antiglobulin reagents are considered polyspecific. And so you guys know this prefix poly basically means it contains more than one thing. So polyspecific AHG contains both anti-IgG and anti-C3 antibodies. Okay, so it could be derived from polyclonal or monoclonal sources. When we see agglutination in our testing, it indicates that IgG or complement is coating the red cells. It doesn't tell us which one, okay, because one of these has bound to either the IgG or the complement. So when we get a positive test using polyspecific AHG, we then have to go on and do a differential test where we test monospecific anti-IgG and monospecific anti-C3 to find out specifically what's coating those red cells, or what is attached there. Okay, so monospecific AHG, it's going to be anti-IgG or anti-C3, but not both together. And so anti-IgG, antibody against IgG, is going to target human IgG like we looked at in the last picture. Anti-C3 is going to specifically target C3, human complement proteins that result from activation of the classical pathway. And this can tell us intravascular hemolysis or extravascular hemolysis is happening. All right. So hopefully you're okay with that concept. Okay. So just remember, antiglobulin reagents are tools to help us determine when sensitization has occurred, and we may not be able to visualize it. So it just allows us to see it. All right, so we're going to get into a certain type of antiglobulin test called the DAT, direct antiglobulin test. The direct antiglobulin test is used to find out if IgG or complement are bound to a patient red blood cells in vivo. So what this means is something has happened in this person, and antibody or complement are bound to their red cells in vivo. We're not doing anything in our test tube. We just want to find out if this sensitization has occurred in vivo in this person. Okay? So we add AHG reagent to our patient's 3 to 5% red cells after they've been washed, and that's it. We don't add anything else because we want to see if these red cells have something attached to them. Okay? It's very important to do proper washing whenever we're using anti-human globulin, because the washing removes any unbound antibodies and complement proteins that might be present in the patient's sample that could interfere with our testing. And a little bit later, we'll look at how they can interfere. So when we see agglutination, it indicates IgG or complement is attached to the patient red cells. Okay, so the important thing to remember for the direct test is sensitization. Immune complexes have formed in vivo. Okay, it's not something we're doing in our test tubes to make this happen. Okay, it's already occurred in the person. Okay, so maybe looking at this picture is going to help a little bit. We have our washed patient red cells, and this is what they look at. This is what they look like before we even add anything. So we see this patient's red cells has antibodies attached. So some type of sensitization has happened in vivo, okay? And so when we add our anti-IgG to the washed cells, represented by the gray, look what happens. Okay, it's kind of like our last picture. This antibody against IgG says, hey, here's IgG, that's my target, I'm going to bind to it. And we get this cross-linking here and lattice formation, and we're able to see that this happened, okay, in the individual. This is a picture I found on the internet, which basically it's just another diagram or a way to look at it. And so once again, sensitization, we see here the antibodies attached to target antigens. This happened in the patient. When we add the anti-IgG, it's allowing us to see that this has already occurred. Okay, down here there's a picture that represents what would happen if complement was coating the patient's red cells. So complement is represented by these little triangles here around the red cells. So when we add antibody against C3, okay, now it says, hey, here's C3, human C3, that's my target. And same thing, it binds. We get our cross-linking in, we're going to see agglutination. It's going to tell us, okay, that it basically happened. Just remember, this has both happened in vivo. Okay, very important to remember. So what can cause sensitization to happen in vivo, resulting in a positive DAT? Okay, let's look at these examples. Somebody had a transfusion reaction, okay? Somebody was given a transfusion of donor red cells, okay? And so basically the donor red cells in the recipient has an antibody that's directed against some antigen on these donor red cells. And so their antibody said, hey, that's my target, and linked onto them. So that sensitization happened in vivo. That's a transfusion reaction. It could be due to hemolytic disease of the fetus and newborn, which we'll spend a lot of time talking about later, okay? So in this case, you know, the mother is carrying the baby, but the baby has different red cell antigens than the mother because father contributes to some of those as well, right? So in this case, the fetal red cells are coated with IgG. So remember, IgG is the class of antibody that can cross over the placenta from the mother. So if mother lacks an antigen that the fetal red cells have, and she's been previously sensitized to that antigen, has an antibody, boop, can cross over and link onto the fetal antigen. So sensitization has happened in this case in the fetus, and we'll talk more about this in a lot of detail later. It could be due to autoimmune hemolytic anemia. Okay, and so patient has an autoantibody, antibody directed against self, and so there could be IgG or complement on the patient's red cells in this case. Or last time, drug-related mechanism. Certain medications in certain people can cause immune complexes to form with that drug. And so this complex of IgG and drug can attach to the person's red cells and cause sensitization. So very important to remember those 4 times that cause the DAT. And just remember, this is happening in vivo in the person. So it's not anything we're doing in our test tube to make happen. It's already happened. All right. So we're just going to do a little bit more, and then I'm going to take a break from this. The indirect antiglobulin test is something that we use when we're doing testing in the lab to detect immune complexes or complement bound to red cells. So the big difference here in vitro, not in vivo, in vitro, in the test tube. So this is something we do to enhance weaker reactions which may not be visible to us initially. Okay, and so it's used as part of another test. So it can be used as part of an antibody screen or part of an antibody panel test that we're doing. So here it's a 2-step procedure. Okay, so we basically are adding an antibody source with a red cell antigen source and incubating them together at 37 degrees. Now, if this is a target for the antigen, sensitization is going to occur, but we may not be able to visualize it if there's not cross-linking. So we wash the mixture, we add our AHG, and then our AHG is going to allow us to see that sensitization occurred. So it's the same idea, it's just we're doing this testing in vitro, okay? So maybe we have our patient sample that has some IgG antibodies, and we're adding our patient's serum or plasma with some type of red cells, maybe screening cells, and we're incubating them, and we see that the IgG sensitization has occurred. However, it's not, we can't visualize this as agglutination because there's no cross-linking going on here. So we do a wash step to remove any unbound proteins, and we add our AHG, anti-human globulin, anti-IgG, and lo and behold, what happens? Remember, this is antibody against IgG. It says, hey, there's my target. There's 2 green IgGs, and we see this cross-linking, and now we're going to visualize that agglutination and be able to see that this happened. Okay, so this is simply something in our toolbox to help us detect weaker reactions that we may not be able to visualize. So when can the indirect antiglobulin test be done? As part of antibody screen, as part of antibody identification with the panel, in a test called a cross match, which we'll learn about later, which is where prior to a transfusion, we combine the patient's serum or plasma with donor red cells that we're thinking of transfusing in them to see if they're compatible. And also an antigen typing, okay, for specific red cell antigens that may be present on patient or donor cells. And so I know that it's a little bit confusing with the DAT and the IAT, and so I did find a YouTube video that I really think kind of sums it up. And so I'm going to stop this share and try to show you the video. It's only a couple minutes long, but if you're not able to hear the video, then you can look at the link separately on your own. Okay, so I'm just going to share a different screen with you guys. And I know sometimes it, you know, my for whatever reason you can't hear well when I try to show you videos, but I'm going to try. And if you can't hear it, you can just look at it on your own. Okay, so not sure if you guys could hear that or not, but if you can't, you could listen to it on your own. I think it just kind of sums things up pretty nicely. So I'm going to go back to our PowerPoint. And we're going to move ahead. Okay. This is a nice summary also that I found on the internet that basically compares and contrasts the direct antiglobulin test to the indirect test. And when you're looking at these, you see that they call them Coombs test. Basically, Coombs was the scientist who came up with the bright idea of how to get antiglobulin reagents. So he's basically the guy that figured out, hey, if I inject a different animal with human IgG and human complement, that animal is going to make an antibody against it, and I can use this to help me find these reactions. And so sometimes you'll hear people call it a Coombs test. And so just remember, direct test, we see that antibodies are already coating the patient's red cells. And we're just using our antiglobulin to help find out that this already happened, okay? In the indirect test, we're adding our patient's serum or plasma that has antibodies with some type of red cells. We're incubating them, and sensitization may occur as in this step, but there's not cross-linking, so we're not able to see or know through agglutination that it's occurred. So by using the anti-IgG, we now see that it's binding to its target IgG. We're getting the cross-linking and we're able to see it. So it's a pretty picture, I think. Maybe that will help. Okay. So I'm going to take a break and stop now, and I'm going to continue this recording and pick up in a little bit with the next part, talking about check cells or Coombs cells.